Now in this topic we are going to see about what are all the changes during muscular contraction happens, right? What are all the changes in muscular contraction? The first changes will be your electrical change. The next we'll be seeing about how the physical changes, how the physical change. Then we'll be seeing the histological change of the muscular contraction and some other chemical and thermal change. Chemical and thermal change. All of the changes are important. In all changes you will be having some different points to remember, but in this the very important changes which I have to highlight for you people is electrical change is a very important topic. At least you have to be attentive in that, and the part of a histological change part where we'll be seeing about excitation contraction coupling. That is important. Excitation contraction coupling. Along with that, a sliding mechanism of our muscular contraction is also there. That is also important topic. Already always the questions have been asked in the topic as well. Okay, yes. While speaking about electrical changes of your muscle contraction, we have some potentials, and because of that electrical potential, how these potential comes into the picture, that is what we are going to see. In that, the first part we are going to see is our RMP. What is RMP? RMP is resting membrane potential. Resting membrane potential. Yes. So then we will be seeing other potential like action potential. How does it works? From the resting membrane potential, how does our action potential of our muscle fibers get starts? Then we will see some difference about what is action potential and the graded potential when electrical changes happens in muscular system, right? So starting with our RMP, in our resting membrane potential, please remember we have to see the balance changes between our ions present in ECF and ICF, so that it will be easy for to understand what is the basic components why we are reading about RMP. It's a potential difference across cell membrane. It's a potential difference across cell membrane, okay? Especially inside and outside of the cell. What we said, especially your ECF and ICF. You can say ICF and ECF. This difference between them should be followed in a balanced way, so that ionic basis will be done, so that your ions will be in a resting membrane potential. Okay, in this basis we need to balance between one. First is your sodium potassium pump. Sodium potassium pump, we can say it as Na plus K plus pump, and then we need a basis between our selective permeability of cell membrane, right? So we have to be very clear between these two things, so that we'll be able to understand how this resting membrane potential is maintained by our cells. So one of the two reasons why the resting membrane potential is maintained in a balanced way, so what will be that? Our channels are there. For example, if this is your ions, this is your ECF and this is your ICF, right? So always remember, our potassium channels are called as a leaky channels. So once if the potassium channels, because of the more positivity, more potassium leaky channels inside them, with this leaky channels potassium tends to come out. Because of this coming out of the potassium with the way of a leaky channels of potassium, then our ions starts to depolarize. But in this way, when the potassium comes out, our ionic basis of all the cells gets disturbed. Just because of the potassium efflux from our leaky channels, positivity will be coming out. So as such, this potassium is having a positive charge. Your positivity in ECF will be more now, as such potassium is effluxed a lot. So positivity outside the cell is more now, or else you can say it's increased, and negativity inside the cell is increased. So this negativity inside the cell makes our potential to be in a membrane called as a resting membrane potential. Simple, okay? Resting membrane potential. So that is important, how our cells are maintained in a form of a resting membrane. So same like for our potassium, they have leaky channels, and our anions like proteins and the ions like a negatively charged substances have also have a channels, and that channel is nothing but our channels for anions. They are also have channels for maintaining them to be in a inside of negatively charged ion, so that it maintains a resting membrane potential. So why our cell is maintained in a resting membrane potential? These are the two reasons. First, because of potassium efflux, which makes positivity outside and negativity inside. That is the reason why RMP is maintained. So this is what all about you have to know in RMP part. The next about what is your action potential. So we have seen how the membrane is in a resting position. Now we are going to see how it acts so that action potential of our muscle gets starts. So main important thing, how the action potential starts, the two phases we have to be very clear of. One is your depolarization, and another one is your repolarization. So what is action potential? Actually it is a electrical change in the membrane. Whenever the nerve is stimulated, you have some electrical potential that is mentioned as action potential. We are going to see it in a two ways. One is your depolarization, another one is your repolarization. So in depolarization, what will happen? In the initial phase, the action potential inside the muscle becomes positive, right? So inside you have more positive cells, and outside actually you have a negative cells. So this is abolished in a result of a depolarization. So inside there will be a positive cells, and outside we have a negative cells. This is important. So what about repolarization? Same, it is also a phase of action potential, but which reverses back to the resting membrane. This repolarization means they reverse back to the resting membrane, right? So once they get reversed back to the resting membrane, within a short time after depolarization, inside becomes negative. As such, it's reverting back to a resting membrane potential. We know what happens in resting membrane potential. Inside it becomes negative, and outside of the cell it becomes positive. So that is important, means inside it becomes more negative, as such K plus have been effluxed, so outside it becomes more positive. So that's why it is reverting back to a phase of resting membrane potential, which shows your depolarization and repolarization. If we are predicting the curve in a repolarization and depolarization, we will get a curve which is like this. So this is the curve predictor which is appreciating the depolarization and repolarization part. So if we can able to see this curve, here is our resting membrane potential RMP. The cell is in a resting potential where inside we have much negativity, and outside of the cell in the ECF we have more positivity, and the muscle is ready to undergo depolarization, but which is not depolarized yet. So while starting the function of depolarization, in between they have some period that is called as a latent period. Now then our depolarization starts. Okay, this is our depolarization. Once the cells are getting revert back to their resting membrane potential like this, they are reverting back. This is termed as your repolarization. So that is important. Again, we'll be having something called as a hyperpolarization, which goes even more repolarized. That is your hyperpolarization. So this all shows your curves which is implementing the muscle contraction, how the action potential, how the stimulus of your muscle is happening. About your depolarization means how they are making their cells to be very positive outside and negative inside, just because of our leaky channels like potassium is within us, right? So in that way we have to know what is latent period over here. So what is latent period? For example, it is in between our resting membrane potential and depolarization. So we can say the period same, it is like our relative period, refractory period. So it's a period where there is no change in the potential. Even the potential stimulus is there will be no change immediately after applying the stimulus. So it will take some time. For example, if a person is pinching you, until and unless if you recognize the person is pinching you, you cannot respond for them. So till the stimulus is there will not be any response for some time. That period, this period is called as your latent period. Then your muscle starts responding for your stimulus, that is your depolarization, which is a starting period of your action potential. So we know what is depolarization, we know what is repolarization. Repolarization, simply you have to remember, in your repolarization you have a spike here. This is called as spike potential. So we said in depolarization, inside there will be more positivity. Why? There will be sodium influx. There will be sodium influx. That's why there is too much of positivity. Again, in repolarization, as we said already, they were reverting back to their RMP. Means inside K plus is going out more, so that outside it becomes more positivity and inside it becomes more negativity. Okay, so sodium influx always denotes your depolarization and potassium efflux always denotes your repolarization. That is important. Now we have been seeing some potential called as a spike potential. So what is spike potential? It is nothing but we have been predicting it in a time of a repolarization where there is a rapid rise will be there in your depolarization part. And that too we have a timing of 0.4 milliseconds. You can see the peak over here. This peak denotes depolarization where it is very rapid, and that too 0.4 milliseconds. Okay, so this sodium influx is also happening just because make them depolarize. So what is happening in the ionic basis? Now we can see, as we know in our RMP, resting membrane potential, we have negativity inside. Why? As our potassium is going out, just because of what channels? Leaky channels. We have positivity outside and negativity inside, right? So this is what happens. Then we'll be going further. That makes our process of depolarization. So what happens in the depolarization? As we know, inside there will be positivity. Why? Just because of sodium influx. Sodium influx by what channels? By voltage gated sodium channels, right? So once they are coming in, then again this is your depolarization. Then now further this depolarization, we know there is a process of repolarization. So what happens in repolarization? We are reverting back to our resting membrane potential. By that way our sodium influx is stopped. So sodium influx is stopped. Then further again potassium efflux will be there, so that they can be reverting back to their resting membrane potential level. So this is happened by voltage gated potassium channels. VG in the sense voltage gated potassium channels. So inside it becomes again negative and outside it becomes again positive. once they are making even more potassium to be coming out. For example, now too much of potassium is coming out. So it becomes more positive outside. So whenever there is a too much of potassium efflux is there, that condition is called as hyperpolarization. Hyperpolarization. This is also important. And once the potassium efflux is stopped, then again sodium influx will be there. This is a cycle of depolarization and repolarization when the leaky channels of potassium makes potassium efflux. So this makes the cycle of resting membrane potential and the action potential, which happens in our depolarization, repolarization, and in our hyperpolarization. So potassium efflux, if it is more through voltage gated potassium channels, again it may cause hyperpolarization, so inside it becomes more negative. So then potassium efflux makes the resting membrane potential, further stimulation will be there. This is a cycle of a muscular contraction where the action potential happens. In these kind of action potential, our scientists predicted some terms about how the deflections are there by a technique where they have predicted the potential terms. What our scientists did after coming to the prediction of these action potential in our muscular contraction, they have recorded the axons response. So axons response have been recorded by a technique where the placement of electrodes outside the axon. So they have placed the electrode. Can you see, able to see in this picture? So the electrodes are placed in the axon from outside, and they have seen a deflection, how actually it works in our muscular contraction regarding the impulse. So first, if you see it in the picture 1, I think you can able to see there is no deflection. Which means there is a zero potential and the cell is in the resting potential, that is RMP. Then what they have did, they have started a deflection. First electrode, they have started a deflection so that it deflects in the left side. Then we can say there is a depolarization within them. This is a picture which is taken from a textbook of Sengbolingam. You can be referring it in a topic of changes during muscular contraction. Yes. The next, see, this picture shows again there is no deflection, which means they are on the way reverting back to their resting membrane potential. See, actually what it happens, if a person is coming and pinching you or pouring a hot water on you, you'll be reacting for that, and then again you will coming to your normal position. You will be not be in excited state, right? So that is what happens. So on the way they are to their RMP, resting membrane potential, again they'll be coming to the repolarization stage. That is what happens here, and you can able to see the graph that it comes down like this. Then again the deflection is started, again it is deflecting more, where there will be more potassium efflux, so that the graph goes down. That is nothing but your hyperpolarization, or you can say this second electrode is working, so that we can say it as depolarization again of the second electrode. Because as we know from the first, we have two electrodes, so then it comes to the normal position, to the baseline. So this shows your recordings of the axons when it was being connected to your electrodes, either it is a first electrode or it is a second electrode, which makes your potentials to be in a recorded manner. Here comes one more potentials while we are recording this, that is nothing but your potential of compound, which we call it as compound action potential. What is compound action potential and what is graded potential? We also have some potential called as graded potential, which is always differ from their action potential. What is it? Compound action potential, see, there is action potential, okay, produced by many nerve fibers. If you are adding all these things, then you will get your compound action potential. That is what which is mentioned. we have to record all the action potential and we have to add all the action potentials. Now, coming to the graded action potential, the graded potential which differs from the action potential, it is a mild change. It is a mild change where your membrane potential is developed, in receptors, synapse, and in your neuromuscular junctions, So this is what important about graded potential. And we also have some difference between your action potential and graded potential, which we are seeing as follows. Yes, now here we are going to see a difference between what is action potential and what is graded potential, and this question and the terms between them are already been asked as an questions in your competitive centralized exams. It is a very easy question, but still if you are not having any idea within them, then we can't able to answer them. Okay, now so see the first thing, what is there? They are a kind of a propagative mode, and graded potential is a kind of a non-propagation, means they have some point of a direction, in that way they'll move. Like in action potential, they'll be in a resting membrane potential, again they'll have action potential of depolarization, then they'll come to repolarization, then hyperpolarization, then normal. So they have some propagation to follow, but graded potential, as it is having a local change within them, there is no direction within them, and they can travel to a long distance. Means, for example, if any part of the body your stimulus is there, it can give impulse to your brain. From the impulse you'll get response even of a sudden from your nerve impulse. Even in you can get it in your graded potential, but only for a short distance. Now coming to the action potential, the next difference is both depolarization and repolarization will be there, but here either you can have only depolarization, or you can have only repolarization or hyperpolarization. Summation is not possible. You cannot add all the things, like you cannot add depolarization plus repolarization, it is not possible. But if you see in your graded potential, you can do that, because as it is a local change, local potential, you can add them, that gives your summation potential. As you know, in action potential we have a period which called as a refractory period, which is having no change, even the stimulus is there, then this starts to get repolarized. But here there is nothing as such a point called as refractory period. And one more important thing, it obeys the law called as all or none law, but it doesn't obeys the law. So this is what the example of action potential and graded potential regarding your electrical changes. Further, we also have some physical changes, which changes regarding the strength or regarding the contractions, which we have seen before in our isotonic and isometric contraction. Now the next important discussion which we are going to have is regarding our histological changes. In the histological changes of our muscular contraction, we are going to see how our muscles get influence themselves and how they are useful in the contraction of our muscle. By the way, there are many steps for it, and main way we are going to see is our actin and myosin function. The next one is your molecular basis of your contraction, in which we will be seeing your excitation contraction coupling, very important topic, and your sliding mechanisms. So these are all already been asked in your exams, and these are all a very important hot topics of the examiner's point of view. It is a conceptual based thing. If you can understand it easily, it is a very easy technique and topic in which the concepts are very important. Even we will be discussing some applied physiology part over here in your excitation contraction as well as in your action potential part. Okay. Now we are going to see how the skeletal muscle gets contracted and what are all the things you have to keep in mind while they are getting contracted. and which histological changes are important though, which we have to remember at least, right? So in this excitation contraction coupling, EC coupling I'm mentioning, first there is an action potential. First there is an action potential in your neuron. So what happens, your friend is pinching you, he is slapping you. Let's take an example, he is slapping you. Further your nerves are there which gives action potential. So action potential in neuron happens. Then what it is there? Again this action potential in neuron causes action potential in your skeletal muscles. Skeletal muscle which causes contraction. So this action potential in your neuron and the skeletal muscle causes a very important function here, that is opening of DHPR. What is this DHPR? Dihydropyridine receptor, which is present in your muscle. So once this dihydropyridine receptors are opened, then this further opens the receptor which is known as ryanodine receptors. So we have seen, already discussed in our cell physiology part, what are all these receptors and what is the importance of these receptors, right? So our sarcoplasmic reticulum is very important for this receptors and the functioning of them. Further opening of this ryanodine receptors gives calcium efflux. From sarcoplasmic reticulum. Okay, sarcoplasmic reticulum. Then once it is opened and the calcium comes out, this calcium binds with our important troponin. What is that? Calcium binds with troponin C. C for calcium, please remember troponin C. Once it got binded, what happens? This role of binding the troponin C along with the calcium, which is what we are going to see in the role of actin and troponin and tropomyosin. See, once the calcium binds with troponin, what happens? As such, you have to know about the binding between calcium and troponin. Before that, you have to know what is happening in your myosin, troponin and tropomyosin. See, now this is your myosin, which we have seen already. Myosin have a head part and their tail part, right? Head part and their tail part. So in this head and tail part, they have a site. This site is called as ATP, or you can say actin binding site. So this is our myosin, where we have a site of actin binding site. So if you now come to see actin, this is your actin. In the actin you have a site same like myosin, but that site is called as a myosin binding site, right? Once this myosin binding site is there, in that myosin binding site, that is covered and prevented by one of the protein that is called as tropomyosin. So this tropomyosin is covering our myosin binding site. Further, this tropomyosin is covered by again a protein which is your C, T and I, which is a troponin. So this is what I am coming to say. Once this troponin C is binded with calcium, that is what happens over here. You can able to see calcium is binding with the troponin C. Then what does it happen? This makes a lateral shift of tropomyosin. Means what happens? Once troponin C goes off with binding along with calcium, then you have lateral shift of tropomyosin. So how can it goes away? See, Once this goes away like this along with the calcium, troponin C goes away along with the calcium, then the tropomyosin is shifted laterally. Then the process which comes in, then the myosin now here can easily be binded with this actin. Means this actin binding site will be vacant, then the actin and the myosin will be binded easily. So myosin binds with actin. So once it's binded, then what is the thing happens? Then our sarcomere gets contracted. So once this contraction is done, obviously contraction of skeletal muscles will be there. So this is none other than our excitation contraction coupling. This process is called as, this phenomenon of lateral shift of our tropomyosin which causes binding of myosin and the actin is called as excitation contraction coupling. Important. Okay. So this is what you have to know at least. They can ask you any points in this. They can ask you a point of myosin binding site and where does the troponin C binds with. They can ask you anything regarding this. Okay. So this is excitation contraction coupling and the role of your sliding mechanism. How the sliding of troponin C along with the calcium with the lateral shift of tropomyosin makes your skeletal muscle to be contracted. Now we are going to see some applied part of what we have seen about the action potential and the excitation contraction coupling. So if you see, for example, what is lignocaine? Lignocaine is a part of a local anesthesia. And how it works, for example, what is the action of this lignocaine in this? They inhibit neurons. Especially while the neurons are inhibited, they causes blockage of sodium channels, isn't it? Obviously, while neurons are inhibited, action potential is inhibited. So blockage of sodium channels will be there. So once this sodium channels are blocked, then what does it happen? Then obviously no depolarization. So no action potential further. That is why no response for your muscle. This action potential makes insensitive of your fibers in the local place. That is how this local anesthesia called as lignocaine is worked. So this is a kind of a applied part. Then one more applied part which we are going to see now is epileptic drugs. Now how the epileptic drugs work? Any ideas? See, what does the epileptic drugs do? They also block the sodium channels. But so these epileptic drugs will make the blockage of sodium channels. Why? Basically, what does the epilepsy mean? There will be too much of excitation of muscle. So too much of excitation of muscle fibers, right? So when these too much of excitation happens, we have to make them inhibition. So this inhibition makes the blockage of sodium channels. Once these sodium channels are blocked, if you see, I am blocking the sodium influx. So what does it happens? Further, once the sodium influx is blocked, potassium efflux is started. Why? As such they are leaky channels. Once this potassium efflux is started, obviously we will know that inside there will be more negativity, outside there will be more positivity. This is a kind of a resting membrane potential. So the thing what we have to achieve, the target what we have to achieve is, the patient or a person who's having epilepsy, he is being excited more, by the way. So that's why our epileptic drugs which we have been giving is blocking the sodium channel, so that potassium efflux have been started then the patient is coming under a potential of resting membrane potential, which makes the patient to be calm and in a normal way. This is what happens in your epileptic drugs. Okay, so now we have been seeing some excitation coupling contraction, right? So what happens in the excitation coupling contraction if any abnormalities are there? We are going to see a second applied physiological part. The first thing which we have seen about the lignocaine and the epileptic drug. The second thing, what happens if suppose there is hypocalcemia? We know that calcium is a very important needed thing for our excitation coupling contraction and also for our skeletal muscle contraction. So what happens in our hypocalcemia and hypercalcemia? Any ideas? Yes. In hypocalcemia, for example, what do you think whether the action potential is affected? See, hypocalcemia means in ECF calcium levels are less. So we call it as hypocalcemia, right? So this hypocalcemia, calcium has usually affinity to sodium. Once there is no calcium in ECF, calcium is less in ECF, sodium are easily capable of coming inside. So sodium is easily influx just because affinity of calcium is less now. So once the affinity is decreased, sodium influx is there. So obviously action potential of neuron will be there. And there is no problem in our skeletal muscle contraction as well. So anyone can ask you a question, hypocalcemia can lead to. So please don't answer if calcium is not there, contraction will not happen. As such, the affinity is decreasing. Sodium is actually doing their work of excitation, action potential. So the skeletal muscle of contraction has never been affected. Question. Okay, this is asked already. Now, to the same, if we are going to concentrate about our hypercalcemia, what is going to happen? So in hypercalcemia, we know that in ECF there are more calcium, right? In ECF there are more calcium. Once there is more calcium in your ECF, calcium efflux will be there, right? This calcium efflux will be automatic in nature, so that concentration of calcium in ECF is higher. So once calcium is efflux more, then what will happen? Easily skeletal muscle are also contracted. So can you able to appreciate the difference between the hypocalcemia skeletal muscle contraction and the hypercalcemia skeletal muscle contraction? So this skeletal muscle contraction further makes too much of calcium influx. So this is what happens between your hypocalcemia part and hypercalcemia part. Sometimes this calcium concentration increase will have no effect in action potential. As such, affinity of sodium and calcium is there. Clear? Now we are going to see an applied part which is called as malignant hyperthermia. What is malignant hyperthermia and why it happens? Please remember, malignant hyperthermia always happens when there is a due to defect of ryanodine receptors. We have seen ryanodine receptors in excitation contraction coupling, right? So when there is a defect of ryanodine receptors, so what happens? There will be automatic efflux of calcium. Automatically calcium comes out from the receptor. So once it is coming out, it is automatically binds with troponin C. So there will be more contraction. Skeletal muscle will be contracted more. So once it is contracted more, automatically in a continuous manner, they tend to produce too much of heat, because there is no one to control the calcium, right? As such, ryanodine receptors are defective. No one is controlled to having a power to control the calcium, so that there will be hyperthermia. Hyperthermia, this is called as a malignant hyperthermia, which means without control this hyperthermia is happening. And remember, the treatment for this is IV dantrolene. Question. IV dantrolene. Question. This has also been asked already. Please remember. Okay, so this malignant hyperthermia, the reason of this is due to the defect of ryanodine receptors. Now we are going to see an important differentiation between your excitation contraction coupling and the sliding mechanism. So both have been in a concept of same, but still some differences are there. What is that difference? Yes, in these two things, in our excitation contraction coupling and the sliding mechanism, both tends to be working for a contraction of a muscle fiber, right? That we know that. But during action potential, the action potential will be happening via, and the calcium is coming via T tubules. Please remember, action potential happens and this makes calcium release from T tubules. Remember, this is one of the important difference between your, which makes the sliding mechanism to be different. In sliding mechanism, whenever the mechanism of lateral shift of the tropomyosin and the myosin head acts, this change in position of troponin and the binding of calcium ions, the release of large amount of calcium from, large amount of calcium is from, especially from the L tubules. This is the important difference I wanted you students to understand. So please make the difference between them. What is, both have been leading to contraction of skeletal muscle. See, both have a same function, which means they have to contract the skeletal muscle. But this is the difference between them. Now, coming to the contraction, we have seen how the muscles are contracted. So now we are going to see how the muscles are being going to relaxed. So the relaxation sequence are, we know that now the muscle is in a contracted stage, right? Now we have to relax the muscle. How the muscle is getting relaxed? We just make the pumping of calcium ions again into the L tubules, okay? Into the L tubules. So once it is pumping in, there is decrease in your calcium content of sarcoplasmic reticulum, because they are pumped into the L tubule. So the sarcoplasmic reticulum content of calcium is decreased. So again, there is a less content and less concentration of calcium. They release the calcium. Who release the calcium ions? Our troponin C releases the calcium. So once they have been releasing the calcium, they detach the myosin head. So already which has been attached with our actin, detachment of myosin head from our actin. This is important, and again the muscle is relaxed. Muscle gets relaxed. So this is the sequence which occurs in our relaxation of muscle. Clear? So what is happening over here? You have to be very confident. Pumping of calcium into the L tubules makes the content lesser in sarcoplasmic reticulum. Once it got lesser, then the release of calcium from the troponin C happens, which makes further relaxation. So this is the important point which we have to remember over here, right? We also have a chemical changes in our muscular contraction. In a chemical changes, we have some pathway. Where we have to see how the chemical change of muscular contraction makes the energy influence. So during the chemical changes of your muscular contraction, so energy and the chemical changes during muscular contraction, especially for the supply of energy, right? During this muscular contraction, we need some energy. So the supply of energy will be happen only by the breakdown of ATP. Only by the breakdown of ATP, right? So this is what happens when the muscular contraction is done. ATP will be broken down. So ATP will be giving ADP plus a phosphate where ADP will be giving a kind of a energy, right? So this is what happens. So while energy is liberated, the muscular contraction have been done. Again during the muscular relaxation, further the how resynthesis of ATP is done. So the resynthesis of ATP is done by many of the process, but one important process which have to be mentioned is by a help of a Lohmann's reaction. By a help of a Lohmann's reaction. What is Lohmann's reaction? Actually, our ADP, which has been given as an energy, this ADP have been combined with CP. What is CP? It's not Connaught place. Please remember it is creatinine phosphokinase. Creatinine phosphokinase. Okay. So once this CP is being added with our adenosine diphosphate, which was released in the form of energy, again we need our ATP. So it was being released by ATP in the form we gained our energy. We resynthesized our ATP along with the formation of creatinine. Okay, that is important. So this creatine phosphokinase, which elaborates the creatine, makes our Lohmann's reaction. Please remember it's creatine phosphokinase. Now coming to the resynthesis of ATP. Now our ATP is formed, energy is synthesized. Even we can resynthesize our ATP by the form of carbohydrates as well. Resynthesis of ATP is also done by carbohydrate mechanisms. How? We can synthesize our carbohydrate by our pathway called as a glycolysis pathway. What is glycolysis pathway? Especially it was being done with Embedden Meyerhof pathway. Clear? So this is also important. By this way we can gain our ATP which is lost back. Also we can gain energy by our Cori cycle. You will be discussing everything in your biochemistry part, but still just get a knowledge by be familiar with those points that yeah, this is the thing where we can gain our resynthesis our ATP further. So this is what we can get during glycolysis Krebs cycle. How much is the part of total ATP we can get? We can get totally 38 ATP. By how? During glycolysis you can get only 2 ATP. During Krebs cycle, you can get again 2 ATP. By utilization of hydrogen in their process, again you can get 34 ATP. That is none other than totally you will get 38 ATP. So this is what important to remember about our resynthesis of ATP. Even you can feel some changes in their pH as well. During muscular contraction, there will be thermal changes also. Okay, thermal changes are also there in muscular contraction. So what are all the thermal changes? Might be they will be in the form of heat, right? There is a resting heat, initial heat, and your recovery heat. So this is what happens in the thermal changes, right? So whatever muscular contraction have been undergone, whatever heat have been produced, we name it as rest, initial and recovery heat. In rest heat, the heat which is produced while you are sitting. Always if you are in a sitting mode, you contract your leg muscles, and if you are sitting in a chair, that also produces heat, right? That is your resting heat at rest. Now about your initial heat have 3 steps in them. While activating your muscular process, there will be a heat. While shortening your muscular process, muscular contraction, there will be a heat. and by relaxation of your muscular process there will be a heat production. That is what gives our initial heat. Recovery heat is nothing but at the end of the activity. Once your muscle is contracted, again they will be relaxed and the process got end up. So this end of the muscle contraction and relaxation have some form of a heat, that is your recovery heat. So this is about the changes in your heat.